There is a new paper called BEAD. That stands for Bridging the Gap Between Experiment and Artificial Intelligence-Based Drug Design. And it comes out of China Resources Pharmaceutical Research Institute in Shenzhen and was posted as a preprint in July 2026. And the reason I picked it is that it puts a very sharp answer to the table on the question I asked a number of times. What is the human actually for once the machine can both think and do? So let me frame the problem the way the authors do. On one side, you have AI-driven design. It can search an almost unimaginably large chemical space, but it has three well-known bad habits. It behaves like a black box, so you can't see why it proposed a molecule. It falls apart when there is very little data, which is exactly the situation for new or rare targets. And it happily draws molecules that are beautiful on screen, but nearly impossible to make in the lab. On the other side, you have the human medicinal chemist. The chemist reasons from precedent, from things that have worked before, and from intuition. That makes their design interpretable and chemically sane. But one person can only hold a tiny sliver of the world's chemistry in their head. And there are more than 17,000 biological targets that have been studied. No humans know them all. So you have breadth without judgment on one side and judgment without breadth on the other side. So BEAD is an attempt to bolt those two halves together. Now here's the part I think we need to be honest about, because I think it's where a lot of these agentic AI papers oversell themselves. The architecture is not exotic. It's a large language model, GPT 4.1 in this case, sitting in the middle as a kind of dispatcher. Around it, they've wired the standard toolkit. Search a chemical database, compare structures in 3D, run docking, predict properties, generate new molecules around a fixed score. The chemist talks to the whole thing in plain language. The clever part isn't really the machinery, it's the workflow they run through it. Most AI drug design goes pick a target, unleash a generative model, get 10,000 novel molecules, then desperately try to filter them. BEAD runs almost the opposite loop. You start from a molecule that's already known to work. You pull out the useful pieces of it, then, and this is the genuinely new idea, you go looking at completely different targets to find cases where chemists have already swapped out the scaffold while keeping the important 3D shape. You borrow that trick, you bring it back to your target, you generate around the new scaffold, you make a handful of molecules, you test them, you see what broke, and you feed that failure back in as an explicit rule for the next round. That is much, much closer to how an experienced chemist actually thinks. So let me sit on that new idea for a second, because it's the intellectual core of the paper. Most machine learning asks, what has my model learned about molecules for this target? BEAD asks a completely different question. Where else in the entire history of medicinal chemistry has somebody already solved a problem shaped like mine? They call it cross-target knowledge transfer. And it's powerful precisely for the targets where AI usually fails. If your target is new and data poor, it doesn't really matter, because the useful knowledge doesn't have to come from your target at all. It can come from a completely unrelated protein where someone years ago did the exact structural move you now need. The system mines across those boundaries in a way no single human can. Now, retrospective demonstrations are nice, but they are easy. You already know the answer exists. You're just rediscovering it. So the part of this paper that earns real attention is the prospective run. A live program and a target called PRMT5, which is a cancer target. They start from a clinical compound. The system proposed 3 alternative scaffolds. And here's the number that made me stop when I was reading the paper. In the first cycle, they synthesized 6 molecules, not 600, just 6. Two of the three scaffold families keep strong activity. One of them, compound 1, is roughly as potent as the reference and dramatically more selective. But it brings a new problem, a hard toxicity risk called hERG. And this is the moment the whole philosophy of the paper shows itself. We don't just ask AI, make it better. A human looks at compound 1, points at a specific piece of the molecule they suspect is causing the toxicity, and turns the suspicion into a hard rule. Never generate anything containing this motif. Then the funnel runs. 50 molecules generated, 37 left after the toxic motif is filtered out, 10 after property screening, 3 chosen by human chemist, 3 made, across the entire live campaign from start to a real preclinical candidate. They synthesize 9 molecules total. The candidate is called CRP-276. And CRP-276 is good, not in a firework way, in a real drug way. Against the current benchmark compound, it's about equally potent as the enzyme, but it's several times more selective in cells. It clears the body more slowly in good way, it lasts longer, it's orally available, and that hard toxicity flag has essentially collapsed from around 68% inhibition down to 8%. In an actual mouse tumor study, it matches the benchmark almost exactly. So the headline is not AI invented the miracle molecule, it's more a tightly controlled human AI loop walk all the way from potency to selectivity to toxicity to pharmacokinetics to a live animal result. And it did it with only 9 compounds. Anyone who's worked in medicinal chemistry knows how very tiny that is actually. Here is what I take from it, and it's slightly against the grain. The industry keeps selling generative chemistry on the size of the space it can explore. You know the famous number 10 to the power of 60. This paper argues the opposite. The problem was never too little exploration. According to the paper, the problem is finding the right small neighborhood to explore it. The scaffold, the constraint, the negative filter, those aren't limitations on the AI, they are the most valuable thing the human brings. Constraint is the product, but I'll be a fair critic too, because you should be skeptical too. This is a preprint, of course, and not yet peer-reviewed. It's one successful project, which is a very small denominator. The retrospective benchmarks are set up in a way that flatters the method, and here is the deepest challenge. The paper proves the whole workflow produced CRP-276, but it never cleanly separates which part did the work. Was it the cross-target search, the constraint, the docking, the language model, or the human chemist making good calls at every fork? Almost every underlying tool here already exists, so the agentic AI framing may actually be crediting the LLM. For what is, in truth, a very well-designed collaboration between all tools and smart humans. Which brings me right back to where I started. If you're looking for the moment the human disappears from science, that isn't it. If anything, it's the opposite. The machine gave the chemists reach they could never have on their own, access to patterns spread across millions of structures and thousands of targets. But every decision that mattered, which trick to borrow, which molecule was worth making, what was probably causing the toxicity, was actually human judgment. So maybe the honest way to describe BEAD isn't an AI drug hunter, it's a medicinal chemist operating system. The humans still decide what's a sensible move. The AI just let them make that move at a scale of 1 against the world's entire chemical memory. And if that efficiency generalized 9 molecules to a preclinical candidate, that's a far bigger deal than the next clever tweak to a generative model. So that's the paper. Let me know what you think in the comment, because I genuinely don't know yet whether this generalizes, and I'd love to be argued with.